We actually caught 3 things. Hi, my name is Micah McCauley. I am a senior research scientist in the Department of Physics in the lab of Mark Williams. We're over here in ISIC with a combination dual beam optical tweezers coupled with a confocal fluorescence imaging system from Lumix. And we are using this today to study the structure of the basic subunit of chromatin, in particular the nucleosome. And we're interested in its stability across a number of different conditions, just for the sake of understanding how the nucleosomes function, but also to understand how they are dysregulated during disease. We have added a fluorescent dye onto 2 distinct pieces of this octamer, onto 2 distinct histones. As we pull the nucleosomes apart, we're going to see what happens individually to those dyes, and that's going to tell us something about that stability. All of our samples are loaded into these tubes, starting from my end and heading away. This is what's called a laminar flow cell that enables all of these samples to flow side by side such that they don't cross each other. The second part is our optical tweezers. So with the beads, they're solid polystyrene, but they're coated with this large functional protein. And in between that tethered DNA, on each end is a single biotin. And the biotin is what attaches to this streptavidin. So it has a bond that it will form with that. Once trapped, we're actually able to both video track the center positions of each of those beads, but also to subtly monitor any deflection of the beads from the center of the laser trap, and that tells us the force. I'm moving the bead that's in the red bullseye, and I'm recording the force on the one that's upstream, and if I move it and suddenly force goes up, that's because there's a piece of DNA that, and the tension in that DNA is going up as I stretch it taut. And so what I'm going to do now is direct that up to another channel. And then I'm going to access the third part of the instrument, which is the confocal system. And so you can see that as we take that picture. What we see now is the 2 trapping beads autofluoresce in the blue-green, and then my DNA tether would run across the middle. We actually caught 3 things, and what you're seeing glowing red are a set of nucleosomes, 12 nucleosomes. The idea of this experiment is that I would like to pull on these nucleosomes, pulling and unwrapping the DNA as I go, and so I'm going to do that right now. And then the remaining proteins will slowly fall off, and I want to know, are they falling off in separate pieces, or is the whole structure falling off all at once? Instead of taking this whole image over and over again, I'm just going to take a single line down the center, and so now everything is unwrapped, and I can usually confirm that by looking at the force extension, and I look for these tiny rips that tell me everything has come apart, and then from there I simply wait for the fluorescence signal to go away, and that tells me that these have all released into solution. This is telling me something about the strength of the interactions of the histones with the DNA and also the histones with each other. And by changing the solution conditions, I'm changing that favorability. And this is an important thing within the cell, which doesn't like to have these supernumerous proteins, these histones, unregulated and floating around. So it likes to keep track of all of these things. Understanding how the cell does that is a very important part, not just of understanding the mechanics of the cell, but understanding disease. Thank you very much. We really appreciate you coming down here to check out what we're doing. We're open, and anyone who's interested in using the instrument should feel free to contact Mark or myself. Thanks again. We really appreciate it.